So very good afternoon to one and all present here. I will try my best to keep my presentation as short as possible because I know we have passed the lunch break and everybody must be hungry. So as mentioned, I would be presenting the BCS-based biowaivers, more of a regulatory topic. These guidelines are already existent for now about 14 years. But there are still a lot of confusion on how to use this and how to go ahead when it comes to applying for a biowaiver to get your drug into the market. So the scope of my talk today would be, first I would discuss the whole biowaiver concept, and then with a few examples discuss how to apply this guidance if you are interested in bringing the drug to the market using a biowaiver concept. So to start off with, if we have two pharmaceutically equivalent products, it need not be therapeutically equivalent because the two products pass through different manufacturing processes. It can have different forms of polymorphs. There are various variables between them. So if I have to say that the safety and the efficacy profile of the innovator and the generic are the same, I have to prove the therapeutic equivalence. And that I can do by bioequivalence testing, because therapeutic equivalence not only means pharmaceutical equivalence, but also bioequivalence. And once that is proved that the two drugs are bioequivalent, I can confidently say that the safety efficacy profile of the generic would be same as the innovator product. So that's what we are actually trying to look into by doing a bioequivalence study. Now, what are the situations when a bioequivalence study can be asked from the regulator side? We have two situations, one within the product and one when it's another product. When it's within the product and you want to scale up your whole batch, then you would be asked to prove that your scaled up batch is bioequivalent with the earlier batch. Or if you have made some changes in your product once the product has come into the market. If there are minor changes, you can go through with just some in vitro testing. But if there are major changes, let's say, for example, you had your whole plant in US, you shifted to India because it's cheaper and set up a whole plant and say, oh, my process is exactly the same. But, and the dissolution is also the same. The regulatory authority would say, no, you need to prove that it's bioequivalent because it's a major change that has happened. The other situation would be when you want to approve lower doses. And if it's another product, for example, a generic, after removal of patents, if you're trying to bring a new product into the market, a generic product, then you have to prove that it's bioequivalent because you don't need to give an in-depth clinical data that's required for an innovative product. So bioequivalence study can basically be performed by pharmacokinetic study. That's the gold standard and generally the preferred method. Pharmacodynamic study, that's what yesterday Dr. Bhupati discussed, where in some cases the pharmacokinetic study doesn't reflect the actual final effect. So in those cases, you may have to use a pharmacodynamic study. And then you have clinical trials in special cases like liposomes or nanoparticles, which you may have to use. And the last but not the least, the biowaiver procedure. Now, the first three that are listed here are all in vivo studies. That is, studies done in humans. The last one that we have here is an in vitro study. And as my title suggests, I would be concentrating on the in vitro study. So what exactly is a biowaiver? A biowaiver means that you are avoiding time-consuming and costly pharmacokinetic studies and instead using an in vitro study, in this case, dissolution test, in order to prove bioequivalence between your test and the comparator product. What are the advantages? The most major one, you are avoiding in vivo studies, which are quite expensive. So when you do an in vitro study, you are reducing the cost of your product, plus the time required to bring your product into the market is also lesser. So if you are a generic company who are dealing with competition, you can get your product faster into the market, plus your product will be far cheaper, so more in demand. The biopharmaceutical or the biowaiver concept is basically based on the Biopharmaceutical Classification System. You must have seen this in different forms in various presentations from yesterday and in the morning too. So in short, it's classified based on the permeability and the solubility of the API or the active pharmaceutical ingredient. And on one end, I have the highly soluble and the highly permeable drug in one. And the other end, or the extreme end, I have class 4 drugs, which are poorly soluble and poorly permeable. So on what is the biowaiver concept based on? It says that if my drug is highly soluble, or it's rapid, the product releases the drug rapidly, and the excipients that are present in the product do not affect the absorption of my drug, then I need not prove bioequivalence of a pharmaceutical equivalent compound using in vivo study. Instead, I can do it with an in vitro study. Now this is the slide which shows the timeline of the evolution of biowaiver concept. So if you see here, the first BCS-based biowaiver concept was introduced by the FDA in 2000. I happened to come across an FDA official who informed me that even though this particular guidance was introduced in the year 2000, the companies were reluctant to use it. One, lack of knowledge on it. Two, they felt that if I use this method, maybe if I go to a different country, they will ask me to do an in vivo study. So why go for this method, do an in vivo study, and try to get my drug approved? And then, slowly when it started getting worldwide acceptance, in 2008, close to 14 drugs were sanctioned or given marketing authorization using the biowaiver concept. In 2010, the EEMA recently revised its guidances and brought the new biowaiver guidance into picture. And in 2006, the WHO introduced its own biowaiver guidances. Now, although we have so many different guidances, I'm sorry to say the situation is so there is lack of harmonization and quite some minor differences between the various guidances that are present. Now, because, as I said, biowaiver is a worldwide accepted concept, I would be just concentrating my talk on the FDA, WHO, and the EEMA because that covers most of the ICH countries. One thing that we need to remember, as per the guidances, so far it is applicable only to immediate release solid pharmaceutical dosage forms that produce systemic action. However, sublingual, buccal, and modified release formulations are not part of this, and hence, if your product belongs to this category, please do not apply for a biowaiver, you will not get it. So what are the steps involved in a biowaiver procedure? It basically has 3 steps. The first one is initial characterization and classification of your drug based on the BCS classification system. Then comes the most important step, that is the risk-benefit analysis. Now here, I would like to bring your notice again to my earlier slide that finally the objective of a bioequivalence study is to prove that the safety and efficacy of my product exist. And I want to keep the risk as low as possible that a patient is not affected because of a wrong decision. And last but not the least, we have the dissolution characterization of the 2 products in question. So what exactly is a highly soluble drug? As per definition, if the dose to solubility ratio of a drug is less than or equal to 250 milliliters in aqueous buffer tested from the pH range of 1 to 6.8 at 37 degrees centigrade, the drug is said to be highly soluble. Again, due to lack of harmonization, the FDA has a pH range of 1 to 7.5. Now the question comes, how does this affect? Let's take an example for amodiaquine hydrochloride, an anti-malarial drug which was subjected to the BCS classification. Now, the dose definition that is given by the WHO is also slightly different. It says it's the highest dose strength that is mentioned in the essential medicine list. The World Health Organization lists out the various medicines which are important throughout the world and should be available, and what dose is mentioned there is said to be the best dose. So based on that, 153 milligram is the highest dose, and pH of 1 to 6.8. And if you see, the dose solubility ratio is less than 250 milliliters, so I can say it's a highly soluble drug. Good. The US FDA says it's the maximum dose strength that is marketed. Luckily, in this example, it's also 150 milligrams, but the pH range is 1 to 7.5. And when I take that into consideration, if you see here, the dose solubility ratio is more than 250 milliliters. That means the same drug is classified as not highly soluble. So in this case, in the first case, maybe it's a class 1 or a 3 drug, but in the second case, it's a class 2 or a 4 drug. Now when you come to the EEMA, it says the dose is defined as the highest single dose that can be administered. The highest single dose of amodiaquine that we can give is 600 milligrams. So that means I have to consider 600 for the dose solubility ratio, and in this case, it marginally passes the highly soluble criteria. So if you go to various parts of the world, your drug is being classified in different forms. So this is the problem that you may come across. Coming to permeability, if the permeability of the drug is more than 85% of the administered dose, then it's said to be highly permeable. Here again, the FDA is a bit conservative, and it says it needs more than 90%. And only then it considers itself or the drug as highly permeable. Now, what data can you use to prove that your drug is highly permeable? Any data in humans is acceptable. Now the question is, how do we get this? There's already an innovative product in the market. They have in-depth clinical data. You can pick that up and find out if the absolute bioavailability is more than 85%. You are on the safe ground, and you can submit it as a supporting document. You can also use permeability studies which were done on CACO2 or MDCK cell lines, but these are only secondary data, and you need to use important references which are highly permeable and poorly permeable as standards to prove that your drug has high permeability. So once you have classified or found the solubility and the permeability of the drug, you can classify them in one of the 4 groups. So now, where do we stand? If we have a class 1 drug, you are on the safe area. But remember, the green doesn't mean that you will get a biowaiver. It only says that your drug substance is an eligible candidate for a biowaiver. It just passes step 1. If you are class 3, you will get a biowaiver if it's the EEMA or the WHO jurisdiction, but not in the FDA. That is presently, we have been hearing for years that the FDA is trying to renew their guidances, but unfortunately, we don't have anything new. If it's a class 4, no go. You will not get a biowaiver. If you have a class 2, only the WHO allows a biowaiver, that too for weak acids, not for bases. So all the new chemical entities that are now being discovered are out of the list because most of them are class 2 drugs. Now going ahead, when you do the initial characterization of the drug, you need to be careful when you come across such situations. The first one, if you have different derivative of the API in your test and the comparator, let's say you have 2 different salts and they show different solubilities. Again, you are talking about 2 different classifications. So if that exists, be careful. So if you're a generic company, try to keep the salt as same as possible is in the comparator drug. If you have polymorphism in your drug, again, there will be question from the regulators. Is this going to affect the efficacy and the safety to the patients? So you have to be sure that it doesn't and you're using the same polymorph as used in the comparator. As said earlier, class 2 and 4 are out of question. If you have reports of bioequivalence in literature available which are not related to dissolution, again, you are on a rickety turf because you will land up with a lot of questions from the regulators. And if your drug is a narrow therapeutic index drug, gone. No go, you will not get a biowaiver because the risk is too high to use an in vitro method to evaluate bioequivalence. Next, as I said, the risk assessment is the most important and integral part of a biowaiver procedure. Now, how can I define risk? Risk is defined as a false positive decision and the ramifications it would have on patient safety. Now, this can happen, or how can this change, or how will the bioavailability of the two products change? It can either be product-based or it can be physiological-based. In product-based, it may depend on the excipients that I use or the manufacturing variable. Let's say I punched a tablet so hard that it is too hard. It takes time to disintegrate, time to dissolve, and the dissolution curves are different. Here, I'm still on the safe side because if something like this happens, I can detect it by my dissolution test. But imagine you add an excipient which influences the gastric motility or permeability of your drug. Let's say absorption enhancers. The effect of this cannot be detected by dissolution. If you do something like this in your product, then the chances of a biowaiver are low because they would say, we are not sure that your product will have the same bioavailability profile as the comparator product. So when you have critical excipients, you need to be careful. The best rule is try to keep them as same as possible as in your innovator product. Now see, one example that I can show here is, here to your left hand side we have primaquine phosphate. I'm sorry it's too small, but the profiles are clearly seen. Here you have the dissolution of the pure drug, the comparator, and the test product. And you see that they all have similar profiles and they're releasing drug very fast. In this case, it's an example of quinine sulfate, another anti-malarial drug. This is the dissolution profile of the pure drug. That means the drug dissolves in the simulated gastric fluid and pH 4.5. In 6.8, the release is very slow. But when we took a product that was marketed in Switzerland, these were druggies, and we did the release profile, it didn't release the drug at all. And when we tested a product that was marketed in the US, that is qualaquine, again, the dissolution profile was quite low. So here you can see that the manufacturing variable actually affects the whole dissolution profile because the drug per se has good dissolution, but once you added the excipients and the manufacturing steps, it affected the dissolution to a large extent. Again, bringing your attention to critical excipients, as I said, sorbitol, mannitol, surfactants, these have a high influence on the permeability of your drug. Hence, as per the EEMA, they say keep these ingredients qualitatively and quantitatively as same as possible as in your innovator product. Once the risk is assessed and you find that the risk is low, that means supra and subtherapeutic drug levels will not drastically affect the patient safety, then you are on a good zone. All you need to prove is the dissolution profiles of your drug is very good. But one thing that we should remember is the advantages, or I would say the risk of a false biowaiver decision should not outweigh the benefits of a biowaiver procedure. If you think the risk is too high, then a pharmacokinetic study is more preferable than a biowaiver procedure. So once that's safe, we move to dissolution. Very simple methods, which is generally used in most of the labs. You either use a paddle or a basket. The volume is around 900 ml or lesser. And then you have to do the test for the comparator and the test product in simulated gastric fluid at pH 1.2, pH 4.5, and 6.8. You should note that you cannot use surfactants in your medium. You can't put that and show that, oh, now the dissolution is very good. That means I should get a biowaiver. No surfactants are allowed. The dissolution, as I said, it's very good to do the dissolution of a pure drug because You get to know what are the problems you may face when you formulate such a drug, and you can avoid it during your formulation strategies. One thing that should be noted is the quality control test that you have for a drug product is not or the dissolution test is not same as the dissolution test that's required for a biowaiver. They are two different tests. So at times it so happens people submit a dossier for a biowaiver with the quality control test that is done in the labs, and they say, oh, it's all matching. But the point is, it's not a biowaiver dissolution test. So you need to go back to your labs and subject the drug and the test product to the same battery of tests as mentioned in the guidelines. So the criteria for dissolution are two. Your drug product can be either very rapidly dissolving or it will be rapidly dissolving. Now, what's very rapidly dissolving? It means more than 85% of the drug is released in 15 minutes. Because class 3 compounds have good solubility but poor permeability, they are very stringent with class 3, so it has to fulfill this criteria. If it doesn't fulfill this criteria, your product will not get a biowaiver if it's a class 3 compound. If it's a class 1 compound, any of the two can be fulfilled. Just that if it's a rapidly dissolving compound, you have to prove that the two profiles are same by doing a statistical similarity test and showing that the F2 value is more than 50. As I said, the WHO allows biowaiver for class 2 compounds which are weak acids, and acid dissolve well at a higher basic pH, hence it should be rapidly dissolving at a pH of 6.8. Once that is done, the possible issue or a hurdle that you can come across is your product dissolves very well in all the three medium, but the comparator does not. If this happens, again, you will not get a biowaiver. You cannot make a better product than the comparator. You cannot make a bad product than the comparator. It should be exactly same like the comparator. So if you come across such a problem, what do we do? The possibility is either locate another comparator because different jurisdiction has different comparators, or use pharmacokinetic studies to do your further in vivo or bioequivalence studies. So to summarize the whole thing, Biowaiver is a surrogate method of evaluating bioequivalence of the generic products or two different products. Just if your drug is a class 1 or a class 3, or if it fulfills dissolution criteria, it doesn't entitle you for a biowaiver. The risk assessment is an integral part, and that needs to be tested. If you have a narrow therapeutic index drug, again, you will not get a biowaiver. And last but not the least, if the risk associated with a false positive biowaiver outweighs the advantages of a biowaiver approach, it's better to go for pharmacokinetic studies. These are some useful links if you are interested on what are the guidances that are put forth by the FDA, European Agency, and the World Health Organization. And I thank you all for your attention and would be pleased to answer your questions if you have any. Thank you.
Any questions?
So you say that you cannot use in any surfactant means you cannot use in simulated gastric fluid?
No.
Only
But these,
background.
yes, because these are biorelevant medias, and they say if it's highly soluble, you do the solubility testing in buffer medium, the aqueous buffer medium. So if they're highly soluble, you should not have problem with your dissolution because of the drug. Whatever dissolution problem you will get is because of your excipients or because of your manufacturing process. Yeah.
Just curious, what's the main rationale for FDA to extend the pH to all the way up to 7.4 rather than 6.8?
Actually, the whole BCS classification was put forth by Gordon Amidon in 1995. And immediately FDA picked it up. Actually, he did the whole research in FDA for the FDA, and that's how they picked up in the SUPA guidelines. So if you see in the timeline, in 2000, when the first guidances came, they just picked up the whole thing and thought, okay, the pH range in the body is 1 to 7.5, and let's pick this range, and it's the safe range. But as as we understood the whole biopharmaceutical aspects of the drug, we understood that most of the immediate release drug get absorbed from the proximal part of the intestine. And the pH at the proximal part is 6.8. So that's the reason EEMA on revision tried to reduce the pH range, because it was more biologically relevant than in the gastrointestinal range than a pH of 7.5. Because if you talk about a pH of 7, you get it right down in your intestine, and if you go to the colon, again you have an acidic pH of 5.5. So presently, as per my information, FDA is, because after 2000 they didn't revise their guidances at all. So presently they are revising, and we think that they would also introduce the BCS class 3 and reduce the permeability and the pH specification same as EEMA. This is what we are expecting, but till it comes out we cannot say much. Yeah.
So does the biowaiver of the drug only depend on the BCS? How about the others, such as the therapeutic index, neurotherapeutic index, or serious conditions?
Right.
For
So
example, amlodipine, you know, amlodipine included in BCS1, but it's used to serious condition. How about that?
So that's what I mentioned, like just because it belongs to class 1 doesn't say that it will get a biowaiver, just that it becomes an eligible candidate. But again, when you say the therapeutic index is low, and if it is one of the narrow therapeutic index drug, then you will not get a biowaiver because the risk associated is so high. That means, imagine if I say this product is bioequivalent and one particular product from it is bioinequivalent. So if a patient takes that medicine, the chances of risk for that patient's safety is very, very high. And in this case, the regulatory authorities are so conservative. That's the reason they are like, no, we don't want to subject it to that risk. In that case, it's better that you go for a pharmacokinetic study and prove your bioequivalence. So just by belonging to class 1 or 3, you will not get a biowaiver.
In vitro, do they have any regulations? In vitro, is it we can go for anything like the other studies, or
Presently, it's the dissolution study which has correlated very well with immediate release solid oral dosage form, because these are for systemic absorption. But as in the first day, what Dr. Bhupati mentioned, let's say for ocular medicines, there are some in vitro tests that can be used. But again, so far the FDA or the EEMA has not shown or put forth a generalized acceptance for this method. Maybe on a case-to-case basis, they may accept it for which particular drug you are presenting it. But I think we are a bit far away to get another in vitro method. But because the science of dissolution we have understood so well, and we have brought it to a step ahead where we use simulation model like GastroPlus and PKSim, by which we can predict or have a good in vivo in vitro correlation. I think so far we are just stuck to dissolution.
Any questions? No more questions. Congratulations, Anita Nair, for his nice presentation. Thank you.
Thank you very much.